Verkerk, a PhD student at the Medical Microbiology Department in Maastricht, the Netherlands. My research focuses on resistant bacteria in the clinical environment, the risks they pose to patients, and the possibilities for surveillance. In this poster, the genomic surveillance of antimicrobial resistance reservoirs in the critical care environment, we show that sink drains are a main reservoir for resistant bacteria. In them, bacteria can persist, exchange their genetic content, and possibly spread to patients. As you all might know, resistant bacteria can be a threat to patients, especially extended spectrum beta-lactamase, also known as ESBLs, and carbapenemase-producing antibacteriales, also known as CPE. So to maintain the spread of these bacteria within the hospital, we identified and monitored possible reservoirs of these bacteria. And then also looked if the sink isolates were related to patient isolates. To investigate this, we collected samples from the environment, including beds, equipment, tap drains, tap aerators, and sink drains, over 12 months. During this period, we also collected patient isolates from routine screenings. We then cultured these bacteria and identified them via MALDI-TOF. They were also tested for the ESBL and CPE production. The ESBL and CPE positive isolates were then typed via whole genome sequencing on the Illumina platform. The in-house pipeline then provided the species, the AMR genes, the plasmid origins of replications present, and clustering information. So, where did we find the main reservoirs? Well, as we can see here in figure 1, where we represent the number of isolates in sinks on the right and non-sinks on the left side, you can see that sinks form the main reservoir and contained over 90% of all cultured isolates from this study. You can also see that most CPE and ESBL isolates were contained in these sinks, as colored in blue. You can also see that over the 6 time points, it remains relatively stable what we found. But do these isolates now also relate to patient isolates? As we can see in figure 2, the phylogenetic trees, that we can see that the patient isolates were grouped together with the environmental isolates based on SNP analysis. We can also see that different resistance profiles shown in different colors were present. This could be only ESBL, only CPE, or both. To investigate these different resistance profiles further, we analyzed the plasmid origins of replications and genetic content of the clusters. And here we observed that the genetic content variation was often linked to the presence of plasmids, but the presence of a plasmid did not necessarily correlate always with that gene present. We also found that genetic content could change because an isolate, a sample, so one sink, where we found 2 different isolates with only 2 months apart, had a change in that genetic content. In conclusion, sink drains form the main reservoir of antimicrobial resistance in the environment of a hospital. In them, bacteria can change or exchange their genetic content, persist, and possibly spread to patients. Of course, I want to thank my sponsors, so ESCMID, for me to be able to present here at IMMEM, and also, of course, the supervisors. Thank you.